Dear students, welcome to the lab session Staphylococci in Bacteriology and Mycology course. In this session, we are going to talk about the main species of Staphylococci, which are relevant to human diseases. Second, diagnostic laboratory test, the differential criteria among Staphylococci species, and how to differentiate between them. Analytical Profile Index, the API or EPI system, ID strip, and then the VITEK Automated Systems, and finally antimicrobial susceptibility test. So what are the main species of Staphylococci? Well, these species can be divided into two major groups, coagulase positive and coagulase negative. Under coagulase positive, we have Staphylococcus aureus. They give golden yellow colonies. And we have also Staphylococcus intermedius, which is non-pigmented. And the intermedius is found in dogs. However, it is only considered when there is dog bite in humans. The coagulase negative, we have the Staphylococcus epidermidis, which gives white colonies, and we have Staphylococcus saprophyticus. They give lemon yellow colonies, and we have the Staphylococcus hemolyticus and Staphylococcus lugdunensis. So these are the clinically relevant Staphylococci species. Well, when we talk about Staphylococcus and especially Staphylococcus aureus, we think about a health problem, which is the nosocomial infection. So what is nosocomial infection? Well, the infection that is acquired in hospitals. So as you can see here in this image, this is in the operation theater, where we have the surgeons and medical staff. They are wearing the face mask. Well, one of the reasons is to stop try to avoid the transmission of Staphylococci from persons who are known to be nasal carriers. What is known so far is that about 20 to 50% of people are nasal carriers of the Staphylococci. And it can very well happen that these Staphylococcus aureus species can go from the medical staff into the patient into infecting the wound. So this is a major source of infection in hospitals, and it is, as we said, it is called nosocomial infection. And also a major health problem associated with Staphylococcus aureus is the antibiotic resistance. And we have MRSA, methicillin-resistant Staph aureus, and we have also VRSA, vancomycin-resistant Staph aureus. So these are one of the problems associated with Staphylococcus. Again, we have other diseases related to Staphylococcus aureus, starting from food poisoning to pimple. This is pimple here. It's hair follicle infection or abscess, and also scalded skin syndrome, 4S syndrome, Ritter syndrome, toxic shock syndrome, endocarditis, osteomyelitis, septic arthritis, and pneumonia. In coagulase negative, well, most of them are opportunistic infections. What do we mean by opportunistic? Well, usually there is no infection, but as soon as the immune system is affected or it doesn't work as expected, then we can see those infections appearing. So one of these, like bacterial endocarditis, and for Staphylococcus epidermidis, they become, for example, resistant to antimicrobials because of their sequestration or hiding in a biofilm. So what is biofilm? Well, biofilm is a very thin layer that is found in medical devices. And in this layer, very thin layer, the microbes will find a hiding place, and this hiding place will protect them from the whatever antimicrobial is administered to those patients. So this is one of the reasons why those patients do not respond well to these infections. And another species we have also under coagulase negative is Staphylococcus saprophyticus. Well, it is known to be associated with urinary tract infections, especially in sexually active young females. So these should be only considered whenever samples or specimens from these group of individuals are collected. So what are these diagnostic tests? Well, if you still remember, we have this bacterial identification flowchart in which we use, first of all, the gram stain to identify 4 different groups based on the reaction, positive or negative, and based also on cell morphology, whether cocci or bacilli. Under cocci, we have gram-positive cocci here. We can see there is catalase positive and catalase negative. So catalase test is very important to differentiate staphylococci from staphylococci and micrococci from the streptococci species. And so after the identification of staphylococci, the next step is how to differentiate between the different species. Well, we have this table, and we have these tests on the left hand, and here are the species. And so as a hemolysis on a blood agar, we can see that we have, this is the hemolysis, the colony here, and around the colony, we see a complete RBC lysis. So this is beta hemolysis. Well, this can be seen in Staphylococcus aureus, but it is variable in epidermidis and saprophyticus. What do we mean by variable? Actually, is that it means it can be either non-hemolytic, or it can be alpha hemolytic or beta hemolytic. So therefore it is variable depending on the species and the strain at hand. Now the second test is the mannitol fermentation. We use for this a special medium, selective and differential medium. Actually, it's called Mannitol Salt Agar. It is mannitol because the fermentation indicates, if it is positive, that we have Staphylococcus aureus. As you can see here, the fermentation is appearing here in the yellow color, and it is caused by Staphylococcus aureus species, while negative in epidermidis and variable in saprophyticus. So variable, it means it can be positive. And the indicator here for mannitol is the phenol red, and whenever there is fermentation by S. aureus, the pH will be acidic, and this will turn the indicator into yellow, from red to yellow. And it is selective because it contains salt at high concentration, 10%, which is enough to select for Staphylococci genus. Now another test is the coagulase test, and this test is always positive in S. aureus, therefore we call it the coagulase positive, if you remember, and it will be negative for epidermidis and saprophyticus. So what is remaining is the saprophyticus. So how are we going to identify this species? Well, novobiocin will be very useful in this case, and if we put the novobiocin disc, as you can see here, that will be here in saprophyticus will be resistant, and it will be sensitive in aureus and epidermidis. Sensitive, why? Because we see this is the disc, and we see here a clear area or zone of inhibition, and here are the colonies. So these are the main tests that are used to differentiate. So this table is very important. Now we go to the test, catalase test, that is usually used to differentiate streptococci, which are negative, from staphylococci and micrococci. And we add, this is a slide method where we put the hydrogen peroxide, and then we add several colonies to the drop. We should have positive and negative controls. In case of positive, there will be elaboration of gas bubbles, and this is all because of the elaboration of oxygen. And by the presence of catalase, the H2O2, hydrogen peroxide, which is a free radical, toxic to the cell, it is a defense mechanism to get rid of the free radicals. and the product will be H2O and oxygen elaborated as gas bubbles, as you can see here in this slide. So this is the positive, this is the negative. Now, coagulase test, we have two formats. The idea of coagulase test is to detect the presence of coagulase enzyme, and the function of coagulase enzyme in the Staphylococci, this will protect the microorganism from phagocytosis, from the immune response. It is one way to evade the human immune response. So coagulase test, actually, it can be present in two different forms, either bound to the cell wall or cell membrane, and this is called the bound coagulase, which is also called a clumping factor, and the other coagulase is the free coagulase, which is released by the cell. Now, the purpose of this test is to differentiate between coagulase negative and coagulase positive. And as you remember, the aureus is always coagulase positive. For the slide method, there will be used here the plasma, and the plasma can be placed on a dry slide, and human plasma can also be used. Here we put a normal saline as a negative control. Then we emulsify the colonies in equation in each drop. We mix and rock the slide for 5 to 10 seconds. Then we check for clumping or clot formation. If there will be clot, then it will be positive. So as you can see here in this slide, here the clot formation is very clear, and this is positive, this is negative. Now for the test, if it is positive, then it is coagulase positive, but if it is negative, we can proceed, or we should confirm the negative result by doing the tube method. This is the tube format of coagulase test, and this is because of the formation of the clot, here it is positive and here it is negative. In the tube method, actually there will be emulsification of several colonies in 0.5 ml of rabbit or human plasma with EDTA, and then incubation at 35 to 37 degrees centigrade for 4 hours. So we look and watch for the clot formation within 1 to 4 hours. If we still have negative result at 4 hours, then we can incubate at room temperature overnight and check again for clot formation. The idea here is that sometimes it will revert. So we don't wait more than 24 hours and read the result, because it can revert to negative. It can go from positive to negative after 24 hours. So it's very important to detect the free coagulase, which is more definite test than the slide method, for between the 4 to 24 hours. And also, as I said, the negative slide method or negative slide result should be confirmed by the tube coagulase test. And here we said EDTA is preferred to over citrate as an anticoagulant. Why? Because sometimes there will be citrate utilizing microorganism. They can give you Staphylococci, they can give you false positive results. So to avoid misleading result, we prefer using the EDTA in this case. Now there is another type or another test which is considered the reference for identification of Staphylococci, which is called the API system, Analytical Profile Index. It is high quality and easy to use, standardized, miniaturized strips of biochemical tests. So this is a biochemical test, and based on different identification databases. This is usually owned by bioMérieux Diagnostics company. It is also considered to be a reference and for identification of bacterial and fungal species. The results after isolation can be reached within 18 to 24 hours. So this is the strip. It is the API strip for identification of staph. And as you can see, these are the small wells. And we have here small wells. And these wells are divided into compartments. So this is compartment 1, 2, 3, 4, 5, 6, and 7. And each compartment is made of 3 small wells. And each well, there are here the biochemical reagents. What we are doing actually is we isolate the microorganism and place the isolate into these wells and do incubation. After incubation, we read the results. The idea and the purpose of the whole strip is to come up with this 7-digit number. The 7-digit number, then we go to the database, we look for that number, and this number will lead us to the species according to these tables. So this is the way of identification. So the idea is how to reach this 7-digit number. Well, here, if we look closely at these wells, we see that each well has a definite weight. Here has a weight of 1, 2, and here is 4. So what are these weights? Well, these are the importance of test. So the test that is more important, this is 4 times more important than test 1. So it is given a weight of 4. Here it is less important, so it will be given a weight of 2. I mean, less important in the identification process for the staphylococci. So if we have the negative results, the result that is negative won't be counted, but when the result positive, it will be added. So in this first compartment, we add the 2 because positive and 4. So 2 plus 4, we have 6. And 1 won't be counted because it is negative. And this is the 6. This is the number, the first digit of this number. Now, if you go to the second compartment, and here are the results, we compare, this is the strip when it is negative, and here is the strip after incubation. So some will turn into positive. So if we go to the second compartment again, we see that it is positive for the first and second, so we add them. 1 plus 2, it is 3, but for the 4, it is negative, so it won't be counted. So 1 plus 3 is 3, and this is the 3 right there. For the third compartment, we do the same, and so on and so forth until we reach to that number. We go to the tables, then that's it, we know the strain that we are dealing with. Good. Now there is another system which is called the VITEK system. This performs all the steps required for identification as well as for antimicrobial susceptibility test, but after the primary inoculum has been prepared and standardized. Why? Because the standardization is very important here. After isolation, we would like to know the turbidity. If you still remember that when you grow microorganisms, you put it in a broth, one of the reasons is to measure the turbidity, and you know the amount of growth. So in order to make all the isolates equal, we have to set them into a certain level of turbidity, and then they are before being introduced into the device or into the system. And the system depends actually not on strips as we have seen in the EPI system, but rather on cards, and each card can hold up to 64 microwells. So these are microwells where the will be fed with the isolate automatically, and these contains either the antimicrobials that are going to be tested in case of AST cards, or the chemical reagents that are required for identification in case of identification. And the system allows kinetic analysis by reading the test every 15 minutes. And usually the optical system combines multi-channel fluorimeter and spectrophotometer and to measure the fluorescence turbidity, and colorimetric signals or optical density. So this is a rapid way for identification as well as for detection of the resistant strains, especially Staphylococcus aureus. Now, what other tests we have? This will lead us to the final topic, which is the antimicrobial susceptibility test. So why it is important? Because Staphylococcus aureus is known to be antibiotic resistant, and it can cause a major health problem in causing nosocomial diseases. So the most classical way of detecting antimicrobial susceptibility is by using the Kirby-Bauer or disk diffusion method. In this method, we use a special medium called Muller-Hinton medium. And the medium will be inoculated with the isolate in question, and will be also antibiotic disc. We put different antibiotic discs or antimicrobial disc here, the white discs, and they are going to be incubated and then overnight, and then next day. First of all, we have to incubate the organisms without the antibiotics. So we require totally 3 days, because in the first day we're going to isolate and incubate only microorganisms, and we inoculate the whole plate until all the plate will be inoculated. So the next day it will be identification for the isolate, and then we put the antimicrobial disc accordingly. And because for certain microorganisms we use a certain set of antimicrobials to test for resistance. And after deciding which antimicrobial disc to be included in our test, then there will be another incubation. And after incubation, we see here some will be resistant, as you can see here, and some will be sensitive. Why? Because there is inhibition zone. This is the zone of killing of microbes. So we have different circles, different diameters. So the result of antimicrobial susceptibility test by using this method, the Kirby-Bauer, will require 3 days in order to give the report of the results. Now, how do we know that this strain or this isolate is sensitive or resistant? Well, we use measurement. This is the ruler where we measure the diameter of each inhibition zone. This is the diameter. And according to tables, which are updated every year, for certain microbes, we know that at this diameter for that antimicrobial, this means sensitive. And for this, so keep in mind that it is not the size. When it appears to you that it is a large inhibition zone, it doesn't necessarily mean it is sensitive. It can be sensitive, it can be intermediate. It all depends on the figure and the number after measurement. So measurement is very important, should be accurate, and it should be compared to tables, database that is updated. According to that number, we know whether this strain is sensitive to that antimicrobial or not. So if it is sensitive, then we are going to use this antimicrobial. The physician will consider that antimicrobial in a prescription or prescribing this for treatment. This is the idea. Now, another method is the latex agglutination test. This method will detect the protein, penicillin binding protein 2A. And this is the protein that is responsible for the resistance in the MRSA colonies, or methicillin-resistant Staph aureus strains, which are the source of a problem. And this protein is encoded actually by a MIC-A gene, MECA gene or MIC-A gene. Why it is superior to PCR? Because PCR will detect only the gene, but here in the latex agglutination test, we are detecting the protein. And as far as the infection is concerned, well, the proteins are usually more relevant to the disease than the genes, because genes can be detected. However, a protein is not being expressed for whatever reason, because of a treatment or because some of gene expression regulation, not leading to the expression to the protein. So usually detecting proteins is superior to the genes when we are talking about diagnostics. And PCR is only detecting the genes. Now, another method is by using a special agar, which is called oxacillin, and this is the oxacillin salt agar, which contains 2.2 to 4% of NaCl, or the chrome agar to detect the MRSA. So this is another way to detect. And here you can see in this image, well, this is the latex agglutination test, and these are the reagents. Another method is by using the cefoxitin 30 microgram disk diffusion test to detect the MRSA. And here, depending on the figure, if we have minimal inhibitory concentration below or equal to 21 millimeter, then this is resistant. If the diameter is more than 20 millimeter, this is sensitive. So again, these figures are important to detect the MRSA strains. Another test is the vancomycin susceptibility test. Here, it can be used to detect the vancomycin intermediate S. aureus, the VISA. They are usually tend to have minimal inhibitory concentration between 4 to 8 microgram per ml. And also to detect the VERSA, the vancomycin-resistant Staph aureus. They usually tend to have MIC more or equal to 16 microgram per ml. Another method is the polymerase chain reaction, as we said earlier, to detect the MEC or MIC-A genes and VAN genes. So these genes are responsible for the MRSA and VERSA strains respectively. And finally, as we said, the VITEK 2 system, which is using the AST card, antimicrobial susceptibility test card for gram-positive cocci, can provide automated and rapid, accurate susceptibility results in as short as 4 hours after isolation. Thank you very much.